Bem-vindos ao nosso espaço, um mergulho profundo nos assuntos mais interessantes de esportes, ciências e temas gerais do momento. Aqui você vai encontrar conteúdo de qualidade, com análises empolgantes e descobertas fascinantes que vão expandir seus horizontes. Nossa missão é trazer informação relevante de forma dinâmica e descontraída. Queremos que você faça parte dessa jornada incrível de conhecimento e diversão. Se gostar do nosso conteúdo, não esqueça de deixar seu like e se inscrever no canal para não perder nenhuma novidade. Ative o sininho também. Aproveite!
If you love science, don't forget to like and subscribe. Imagine you are an archaeologist 50 million years from now, trying to reconstruct the human race based only on our teeth and our glasses. You might conclude that there were thousands of distinct, genetically isolated species of humans, simply because everyone had a slightly different prescription lens. This is the fundamental, hidden bias plaguing paleontology, and it centers on one of the most durable, yet deceptively variable, biological structures: the fossilized lens. We call this phenomenon the optical illusion of extinction, and it poses a serious challenge to modern taxonomy. When scientists study the fossil record, they are inherently limited. They can only classify creatures based on what survives the process of decay and mineralization: hard parts like shells, bones, and, critically, optical structures. Take the iconic Trilobite, an ancient arthropod that ruled the oceans for hundreds of millions of years. Their eyes were revolutionary. Unlike the soft, organic lenses found in most modern creatures, Trilobite lenses were made of crystalline calcite, a highly robust mineral form of calcium carbonate. This mineral structure is incredibly resilient, often surviving intact where the rest of the organism, the soft body, the nervous system, and even surrounding tissues, dissolved into dust. Calcite lenses are, essentially, pre-fossilized. They are so common and so well preserved that they become the go-to metric for identification and species demarcation. But here's the problem: this durability masks immense biological variability. While two different species might indeed have recognizably different eye structures, two individuals of the same species might also show significant variations in their lens geometry depending on their life stage, their habitat depth, or the ambient light conditions they were exposed to. For example, if a trilobite species adapted to both shallow, bright waters and deeper, darker regions, its lens facets might evolve slightly different curvatures or crystalline arrangements to maximize light capture. To a taxonomist relying solely on the preserved hard structure, these optical differences, which are merely examples of intraspecific adaptation or phenotypic plasticity, are often interpreted as evidence of distinct, separate species. This leads to what scientists call optical inflation of the fossil record. We end up splitting what should be a single species into multiple extinct lines. Imagine finding 20 different types of trilobite eyes in the same geographic layer. Instead of recognizing this as one successful species that had 20 different localized optical adaptations, the traditional taxonomic approach catalogs them as 20 unique species. When those 20 distinct species disappear from the fossil record, it looks like a catastrophic extinction event, masking the reality that only one evolutionary lineage was actually lost. This bias fundamentally skews our understanding of biodiversity during specific epochs, particularly the Paleozoic era when such crystalline lenses were common. It makes ancient communities appear far more fragmented and specialized than they might have been. More importantly, it can distort the severity of true mass extinction events. If a major extinction event wiped out 50% of recognized species, but 30% of those were actually just optical variations of the survivors, the real extinction rate was far lower. Modern paleontologists are fighting back against this optical illusion. They are using advanced techniques like micro-CT scanning and chemical analysis to look beyond the external morphology of the lens. They analyze the growth rings within the calcite structure and use comparative optics to model how light would have actually focused in the ancient environment. They are also utilizing machine learning algorithms trained on modern species variability to create more robust models of how much optical variation is expected within a single species. By unifying these artificially split lineages, scientists are beginning to realize that ancient ecosystems might have been defined by fewer, but more environmentally flexible, species than previously thought. This shift not only corrects the historical species count but gives us a truer picture of evolutionary robustness and resilience. Understanding the optical illusion of extinction is crucial. It reminds us that the fossil record is not a perfect history book, but a fragmentary artifact, and sometimes, the best preserved evidence is the most misleading. For more incredible facts like this, don't forget to subscribe and turn on notifications.